Okay, and I guess whenever you're ready, Blairanne, you can go ahead and share your screen. And yeah, Daniel, I was telling everybody, I'm just letting everybody introduce themselves. These are pretty short, so I don't have any introductions prepared.
Sounds perfect.
So, and I guess you can go ahead and start, Blairanne, and I'll stop you at 4:15. You're muted though.
Sorry, when I share my screen, it makes my mute button hide. So can you hear me now? Am I good?
Yeah, I can hear you. Yeah, that's weird thing, especially if you have two screens, then the other screen goes over. Yeah.
Yeah. So when I'm looking to the other direction, I'm looking at the data or at the slides, not you, not ignoring.
Yeah. Yeah.
So my name is Blairanne Williams. I'm at Western Kentucky, and so I work on platinum compounds. I'm not trying to cure cancer. I'm really interested in the structure of these compounds and how their toxicity is related. So because I'm interested in metals and metal transport, that's kind of how I ended up here. So it is related to cancer, though I am myself not a cancer biologist. But we are going to look at how the structure impacts toxicity, sorry. And so you'll see a pretty little slide. This is not my data. These are healthy colorectal cells that just wait for a pretty picture just to start off our talk, since platinum is often used to treat that, or cancer, colorectal cancer. So there are 3 platinum chemotherapeutics in the United States that are approved by the FDA. And as you guys probably well know, that 75% of the individuals who receive a platinum chemotherapeutic will receive one of these platinum compounds. And so these molecules have some similarities, and I have them shaded. So red is the non-leading ligand and green is the leading ligand. And I'll talk a little bit more about what that means in just a moment. You'll see that the first generation of these compounds that was created was cisplatin, and so cisplatin was slightly modified in the second generation. The non-leading ligand group in red remains the same, and then they added this larger, bulkier structure on the leading ligand. And then the third generation oxaliplatin, you'll see that both parts of the molecule have been modified, and so there's a pretty well-established role for the non-leading ligand in affecting toxicity in a specific manner. Less is known about the leading ligand. And so we're going to spend a little bit of time today talking about that. So what is the non-leading ligand and the leading ligand? So when this molecule enters the cell, the leading ligand actually leaves the molecule as equated with water. In the case of cisplatin, where there is not a the ligand's not bound, both of these can come off individually, and so you can actually catch an intermediate molecule with only one chlorine atom bound. In these other molecules like carboplatin and oxaliplatin, where there's this bulky ligand group attaching to both of the binding sites on the platinum, you don't really catch that intermediate. They pop off so quickly together because of the effect of the ligand. So when we think about these compounds in treating cancer, there are some overlap between which compounds are used to treat which cancer type. And so you can see here, these are basically can be thought of as efficacies. And so where these compounds work most efficiently. And so for both cisplatin and carboplatin, they can be used to treat small cell lung cancer or ovarian cancer. Cisplatin is preferred for testicular cancer. And then when we start to have this greatest change in the structure, with oxaliplatin, you'll see that we see this kind of divergent new set of cancers where the efficacy is highest. And so I removed this slide for time's sake, but just to briefly say what happens when all of these leading ligands are removed and water equates, it then travels to the nucleus, the compound, and where the leading ligand was attached, those 2 sites bind to DNA, typically at guanines and N7, and it basically just causes the DNA structure to kink. And then there's transcriptional machinery can't get through. It's p53 mediated cell deaths. And this is fairly well accepted in the field and in literature, that all these bifunctional compounds work that way. And so they might bind at different parts of the DNA, but they have a similar mechanism of death. What's not really understood is how they're getting into the cell, which is what I'm most interested in. There's some evidence that there is energy required, and lots of people have done studies where they look at this specific compound and this specific cell type, but they don't look really beyond the compound interest that they have. And so a lot of data currently is focused on cisplatin coming through copper transporters. There's a paper in yeast where that's been done, and there's some other papers where they've looked in mammalian cell lines of copper transporter knockouts and have seen that there's a decrease in platinum uptake. But I kind of want to, my goal is to kind of expand this literature to look more broadly at how the structure is affecting uptake in the different cell types, to try to explain these efficacy differences that we see, because it's not bioavailability, because we can see them in a cell dish as well as in treatment protocols, which are always done by point of origin of the cancer and not to the tissue where it's metastasized to. And so we have a bank of novel compounds that were designed by our collaborator. In this talk, I'm going to just focus on 3 of those. And so you'll notice that in our compound, we have a different non-leading ligand shown here in red. And they were designed so that keeping the non-leading ligand the same, we could really focus on the structural differences by the leading ligand. And so these are the FDA approved drugs, so you can just see them for comparison's sake. So our platinum-EN chloride compound is similar to cisplatin at the leading ligand. Platinum-EN CBDCA is similar to carboplatin. They have the same leading ligand. However, we have a platinum-EN mol compound that's slightly different. It's kind of an intermediate between carboplatin and oxaliplatin. We do have a compound that we are testing that has the same leading, the oxalate leading ligand as in oxaliplatin, but I didn't include any of that data today. If you have any questions about it, though, I can maybe help you along. So this is a cell survival assay. So this is a metabolic assay. So we have used MTT assay, which basically measures mitochondrial function. And we are looking at cell survival. And so all of this data is normalized to untreated control for each cell types. The colors do carry through. So you'll see like the hot pink color is HT29s, which is a colorectal cell line. We'll talk about this quite a bit today. The blue, we won't talk about a whole lot, but that's small cell lung cancer. The gray is HEC293s, which are non-cancerous, but we understand a little bit about the mutation. They're a fibroblast. And so that gray here and the gray there is kind of buried behind. And then last but not least, we used a melanoma cell line. And so you'll see that in our platinum-EN chloride, we do have a few other cell lines that are not included in this. We just haven't done all of them in cisplatin. So on the left is cisplatin, the FDA approved drug. And on the right is the platinum-EN chloride, which is most like the compound that we have that is most like cisplatin. And the first thing you can see is that for both compounds, there's a dose-dependent response. So as we put more platinum on, the cells die. And you'll also notice there's some similarities in what's more toxic and less toxic. So the colorectal line in pink is a little less sensitive to both compounds compared to, for example, the melanoma cell line. With some differences, though, the HEC cell lines and the melanoma cell lines do have more differences in cisplatin than they do in our platinum-EN chloride. Another thing you should notice is that cisplatin is extremely toxic compared to this compound. And so we measure that by EC50 values, but it's well known that of all the FDA approved drugs, that cisplatin is the most toxic. And for us, that does pose a bit of a problem when we're looking at cell death. If it's super toxic, then we lose all of our materials for studying that. And so it's actually nice to have a compound that's a little less toxic to be able to analyze some of the other factors without losing all your cells. One important thing is you will notice that in a lot of these cases, we see this plateau, even in concentrations where we're not, approaching this lower ends of death. And even if we add a lot more, double the amount of platinum that you see, we'll still see these plateau effects where we just have some population of the cells we don't kill. We're actually kind of interested in that because there's a huge amount of literature now that is looking at platinum resistance and platinum's ability to activate the immune system. And so that's something that in the future we're really interested in kind of chasing, that we haven't chased that very much here. Okay, so this slide is basically the same data that you just saw, but we kind of regrouped it. And what we're looking at now is we're looking at on one graph, one cell type, on the other graph, a different cell type. And so this is the data from the 293 human embryonic kidney cells on the left, and then on the right is a colorectal carcinoma. So this was the pink line here, and the left graph is the gray line that's kind of buried. And so we have graphed 4 different compounds together in this slide. So we have cisplatin, the FDA-approved compound, our platinum EN chloride, the CBDCA, and platinum EN mol. And what you'll see is we still see our dose-dependent effect. And so what you'll also notice is that the toxicity, so again, these are all normalized, but the toxicities are very different in the HEC cells than in the HT29s. Comparing, cell type to cell type, which is what we saw in the previous graph. So we have this concentration dependent and cell type dependent effect. What we don't see here is a distinct difference in the leading ligand and its effect on the cells. And there might be some of these lower concentrations. We can see that cisplatin and platinum EN chloride have some similarities here that are different from the others, but it's not something that we see through the whole curve. But when we look at other cell lines, such as the melanoma cell lines, we do see this effect. So I dropped the cisplatin data from this just because of keeping that leading ligand the same, or non-leading ligand the same. So this is truly just comparing the leading ligand. We can see that here, these are statistically significant. The dose response curve overall from the platinum EN chloride is different from both the CBDCA and the MAO. And so we can see some difference that's due to the chloride atom itself. And so when we start to look at, well, these things are toxic, we know that now, we kind of have a range of toxicity. The next thing we just have to do is to see, is the platinum getting into the cell or something else odd happening? And while we were doing that, and since we're interested in transport, we measured other metals in the cells. So we're going to focus for the rest of the talk on just these 3 compounds and their uptake into just one cell line for time comparison. So this is actually the CBDCA compound. This is one of our novel compounds. And so we used 4 different concentrations of the compound, placed it on the cells the same time point as the previous data, which was 24 hours. And then harvested the lysates and measured the intracellular concentration of iron and expressed it here as amount of protein in the sample. And so this is a small n, it's just an n of 2, but you can see that at lower concentrations, it doesn't seem to be changing. This number is actually being driven by 1 data point. And so you can see the giant error bars. I have a feeling that once we get our n up and the rest of that data stuck in here, that this probably will not be different. Now, this doesn't surprise us. We didn't really expect platinum to enter into the cell through the iron transporter because iron is transported through transferrin, which takes up a +3 molecule, and platinum's typically thought of as +2. And so, but this is really nice because typically when one metal is out of whack in the cell, there's a switch in the other metals, and you can kind of see this homeostasis. So the big takeaway is we don't think that this compound is affecting iron transport or iron levels in the cell more accurately. And this was in the HEC cells. So using the same cell lysates, we also measured platinum and copper. And the reason we split this is because there's so much iron in the cell that it makes the graphs hard to read together. So when I separated the iron and the copper and the platinum just for that sake. And so platinum does go up as we treat the cells. And then you can see that copper actually seems to be going down. Now, again, this is an N of 2, so we haven't run any statistics on this, but we're kind of excited about this. So this does kind of collaborate some of the other data that we've seen. We have other metals that we may go back and add into this. One of the caveats of this assay, so we're using atomic absorption because the platinum and the copper are just concentrations are just very low in the cell after exposure. And so this instrument's more sensitive compared to other instruments that we have access to. So we do see both a dose-dependent effect and a cell type-dependent effect in our cells. We already knew from other literature that non-leading ligand was important, but we seem to have some effect for the leading ligand in toxicity and the cell's type-specific nature of the toxicity. We do see platinum levels go up with exposure, and our preliminary data suggests that iron transport is not affected, but that copper levels may be altered in the cell. And so that idea is that as the platinum is competing with the copper uptake, that the more platinum you put on, the less copper that can get in through those transporters. And so we have the lysates for all cell lines that we talked about today and compounds treated and ready to take to the AA. They're in some state of flux of whether they've been read or not. And so most of that data will come soon. And so we're really excited to see what the rest of that holds. Next, we want to kind of move to looking at the transporters themselves. And so if we see an effect on the copper transporter, do we see both proteins that would be responsible for influx and efflux change with exposure to the compounds? And then, I mentioned earlier there was that plateau in our cell survival data. I'm really interested in that and seeing how pathways that would be activated by metal exposure are affected in those cells versus the cells that are responding to the platinum. And do any of those signaling pathways have components then that will affect things like T cell activation as they come in to clear up the cancers, since resistance to platinum is such an important topic at the moment. So this apparently is going to be my Christmas present from my lab. Someone drew a picture of all of us. And so I just want to thank all of my students that are in the lab. I have a lot of undergraduates. I have 7 currently. 14 total have kind of worked on this project, and 2 graduate students have contributed. But it's a really nice little group of kids, and we have a lot of fun. So that was they'll die when they know I put that on there. So with that, thank you for your time, and take any questions.
Great, thank you. A little over time, but maybe time for one quick question. Anybody?
Actually, Blairanne,
yeah.
This is Mark Barget from NKU. That was a really nice talk. I was just wondering what the concentrations were they comparable to the clinic, and especially we're talking about transport and saturation and so forth. Do you have that concentration? Can you comment on that a little bit?
So cisplatin, looking at its IC50 values for the cell survival curves, is much more toxic. And so, for example, if we look in the hex cells, it's about 5 micromolar. In the platinum E chloride that was most similar, it's around 45 micromolar. So it's quite a bit different. And they're all that sort of level of difference or more. So cisplatin is much more toxic. If you look at something like the oxalate compound and compare it to oxaliplatin, in the hex cells, in our hands, it's about 60 micromolar is the EC50. And then again, I didn't write that down I don't remember what it is now off the oxaliplatin off the top of my head. I thought I wrote it down. That was for the clinical, but our actual platinum E and oxalate, I didn't. It's about 60 as well, I think. No, it's a little bit higher than that. No, it's not going to pop up. So, but they're slightly different. So,
it sounds like you're in the range though, and that's what I was kind of, you like a scale above it, and it sounds like you're hitting the sweet spot there. So, thanks.
All right, thanks, Blairanne. I think we better move on to Ali.
Okay, so I'm going to share my screen. Let me see. Do you guys see my screen okay?
Yep.
Wonderful, wonderful. So, hello everybody. My name is Ali ER. I'm a physics faculty at Western Kentucky University, and today I'm going to talk about methylene blue functionalized silver nanoparticles as a potential photodynamic therapy agent for the prosthetic joint infection. So it's kind of a long title, but hopefully you guys will enjoy it. Well, I mean, let me start with the problem. So, prosthetic joint infection is a serious infection type. Normally, it occurs like on the the 2 to 3% of the surgeries, but once it is detected, I mean this infection is quite serious, and the doctor needs to do the surgery from scratch. And unfortunately for us, that this demand for this arthroplasty is expected to increase by 400% from 2000 to 2030. So this is a huge increase, of course. So the problem is, it causes pain, prolonged hospitalization, and it costs up to $100,000 per case. And we know that multiple organism causes prosthetic joint infection, including gram-positive, gram-negative bacteria, and many other organisms. Well, so there are many different measures were taken to prevent the prosthetic joint infection, such as using antibiotics. However, we know that some of the bacteria develop a resistance against antibiotics, and therefore those bacteria poses a great deal of threat for those who have the surgery. So, given the challenges associated with this prosthetic joint infection, and obviously we need to identify an alternative treatment so that we can prevent PJI. So this is a kind of graphical abstract of my talk. Basically, what we do, we have a laser, and then we focus the laser into a silver, sorry, in a liquid. We create silver nanoparticles, and then this silver nanoparticles later is attached by methylene blue, and then we measure the singlet oxygen, and then from singlet oxygen we use the singlet oxygen into deactivate the bacteria, both gram-positive and gram-negative, and then hopefully we will use this in the real world in the prosthetic joint infection. Here's a small setup of our experiment. We have different light sources. Really, the type of the light depends on the photosensitizer we use, and this is our laser setup in our lab. All right. So, well, I mean, why do we use silver nanoparticles? The problem is, I mean, normally methylene blue is a standard, kind of industry standard photosensitizer. I mean, it is very well known, and then it creates its quantum yield is around 0.54, one of the highest among the photosensitizers. And methylene blue, I mean, works fine. When you look at this graph right here, it works perfect in PBS and in DI water. So those are the sterile environment. So methylene blue, this is its UV absorption. You can see that here it has this the 660 nanometer, which corresponds to red light absorption band. So when you look at this DI and the PBS, basically you can see that it keeps absorbing over the time. However, methylene blue has been shown to be an ineffective bacterial killing agent, especially in the human blood. The bleaching of the methylene blue in the plasma is thought to be caused by an attachment of hydrogen into this electron deficient nitrogen here. So in the plasma we have proteins, right? So we have too many amino acids. So we basically went by each protein. So when we put albumin, we saw that this bleaching is even further than the blood itself. So, and then we looked at the individual amino acids, and then we saw that when we use cysteine, this is my cysteine here, it basically goes to in a couple of minutes, it basically bleaches. This blue, it's a very nice blue color, by the way. I can watch it for hours to dissolve in the water. But this blue color basically becomes, it bleaches. It almost becomes the water. So there's just the limited amount of the water, the blue color there. So it basically means it doesn't absorb anymore. So if it doesn't absorb anymore, meaning it doesn't create synthetic oxygen, and therefore it's not going to kill any bacteria. Now the problem is, this is the structure of the methylene blue here. So this central nitrogen is an electron deficient. So cysteine has a thiol group, and then which is, the thiol is, the sulfur and the hydrogen. So whenever we have this cysteine present, that hydrogen attaches to this nitrogen here, and when it attaches, it basically what it does, it breaks the conjugation, so methylene blue becomes glucomethylene blue, and then it doesn't absorb light, so therefore it doesn't deactivate the bacteria. So that's a problem. Now, to solve this problem, we propose to use silver nanoparticles and with methylene blue, and then use it in the bacteria deactivation. Now, those are some of our results, by the way. So we use the Nd:YAG 1064 nanometer nanosecond laser to create silver nanoparticles, and you can see that there are like the multiple silver nanoparticles here. We have used like different mediums. We want to see which one works better for our purpose. So we have grown in the silver nanoparticles in citrate, PBP, PVA. We have used 532 nanometers, second harmonic. We use second harmonic crystal to get 532 and then 1064. And when we did that, when we look at our results, so for instance here, this is our citrate solution at 1064. We use like the different power from 0.5 to 1 and then to 2 watt. And then as you can see, within this range, our size of the silver nanoparticles didn't really change a lot. So it was like around 6 nanometers. When we use 532 nanometer, our size decreased to like the 4.5 nanometer, which is good, by the way. I mean, this is a reproducible result. So which is kind of, we are quite happy with this, the stability of the size. Now, later we looked at the UV vis spectroscopic measurement. This is basically, so remember, this is silver is a metal, and the metal shows like the color in the liquid, because their electron in the d electrons within that metal can be excited from one electronic state to another state. So the absorption around this 400 nanometer is basically a typical plasma absorption of the silver nanoparticles when the frequency of the incident photon is resonant with the collective electron oscillation on the surface of the silver nanoparticles. So basically what we have seen here, so the small spherical nanocrystals should be expected to exhibit single surface plasmon band, whereas the anisotropic particles should exhibit 2 or 3 bands. It really depends on the size, shape, and the concentration. So basically what we have seen here is, as you can see at the 2 watt, we have this broader absorption. So this broader absorption basically becomes narrower when we use the 1 watt. So this is basically supports on TM measurements. In the TM measurements, when we increase the power, it slightly increases the area size of the particles. So in addition, our silver nanoparticles have more uniform size distribution at 2 watt compared to 1 watt. So, and then we use this silver nanoparticles in E. coli first. So as you can see, this blue one here is the E. coli. I mean, this is our reference point. And when we use silver nanoparticles with the E. coli only, as you see, none of the silver nanoparticles are affected by itself. But here's the thing. I mean, silver nanoparticles are known to show antimicrobial properties. But the wavelength we use here is 660 nanometers. So silver nanoparticles do not absorb at that wavelength. They do absorb, however, in UV range. So whenever we use methylene blue here, so when you look at this, when you look at this black curve here, as you can see, non-methylene blue is used with bacteria. The bacteria number, colonies of the bacteria decreases. So when we use methylene blue silver and the bacteria, this increase, I mean, is even the more enhanced, and then you can see that in 6 minutes basically we don't have anything. So we have used like the 3 different mediums to see which one is best for us. So this is our citrate, this is our PVA, and this is our PVP. So we got the most efficient result in PVP. So we did the similar experiment. With gram positive as well. So this is, again, I mean, you can see that whenever we use methylene blue and the silver. We forgot to put silver here. It is more effective than methylene blue and the silver alone. All right, so this is then SEM images of an E. coli bacteria. This is the first figure is the E. coli before any treatment. You can see that, I mean, they're kind of lively, you know, I mean, that they have this, you know, they're round, good shapes. And then here you can see the absorption of the cellular nanoparticles. They're taken up by the bacteria. And then this is after the radiation. You can see that, I mean, their structure, the cell structure, the wall structure is basically, there's a huge change. And also you can see those cellular nanoparticles is outside, meaning the cell wall is perforated and then it releases the content into environment. All right, so we have also looked at the effect of the size of cellular nanoparticles on the bacteria, and then we showed that when the size decreases, it becomes more effective. The probable reason is that the smaller size is able to penetrate more inside the bacteria. Therefore, when it combines with methylene blue, they do become more effective. So, but we know that the cellular nanoparticles can be cytotoxic. So they do show antimicrobial properties, but they can also be cytotoxic. And their cytotoxicity, by the way, depends on size, surface chemistry, exposure time, concentration, and the method of exposure. And normally, in the literature, we know that the liver, spleen, lungs, and the kidneys are the major target organ for the cellular nanoparticles. Although they are eventually degraded and removed by the body. I mean, they can stay in the human body like up to 6 days. So here, in this graph, basically what we use, this is the maximum amount of the maximum concentration we use in our experiment. So we looked at the cytotoxicity measurement. So in this is the HEK 293 cell. We obtained from the American type culture collection, and then we grown in DMEM, medium with 10% FPS. And then you can see that basically within this range, I mean, they're not cytotoxic. So I mean, it's not really a major issue for our purpose. Okay? All right. So as a summary, we grow the cellular nanoparticles using pulse liquid ablation in different medium. We showed that laser fluence was shown to affect the average size of the cellular nanoparticles. Although this part needs to be clarified a little bit more because the size effect is not really prominent. So we are still continuing to understand this problem. And then we characterize the cellular nanoparticles using TEM, SEM, UV, and photoluminescence. So when we combine the cellular nanoparticles with methylene blue, they do show higher synthesized generation which in turn translates into higher bacterial deactivation. And we were able to deactivate gram-positive and gram-negative bacteria. We measured the cytotoxicity, and then up to 50 microgram per milliliter, our cellular nanoparticles didn't show significant cytotoxicity. And then we also looked at the morphology of bacteria before and after irradiation, and it shows a clear difference in the morphology. So for the future, the very next step will be to do this experiment in the biofilms, which we already grow the biofilms on a HAP, and we are going to grow on the titanium as well. And then later we are going to measure the transient kinetics of the cellular nanoparticles so that we can understand the mechanism. And hopefully this understanding of mechanism will help us to determine the way to improve the single oxygen generation. And then later we will use this technique directly in the synovial fluid for the prosthetic joint infection. So those are our group members. Jordan Andrew works in our group, and Blairanne works with Dr. Banga. So Dr. Banga is our collaborator. She's a co-PI in this project. And then Dr. Badruddin, she's a faculty member in UK School of Medicine. And Somon and Salajans are my graduate student who works in this project. And with that, I'm going to end my presentation. I'll be happy to answer any questions you might have. Thank you.
Thanks, Ali. That was great. I was just curious why it's toxic to the bacteria, but not the HEK cells. I mean, do the HEK cells just not take up the silver particles, or they take them up but it's just not a concentration that would be harmful because it's a larger cell or whatever?
Yeah,
You know
actually,
anything?
Dr. Bang is here. She can answer better than me. But so we have not irradiated it with methylene blue. So we wanted to look at the dark cytotoxicity by itself. So there was not like, the irradiation with the red light. So there wasn't any single oxygen generation. Is that true, Dr. Banga? That's how we did it, right?
Yeah, that's correct. So this cytotoxicity was mainly for the silver nanoparticles. We were just trying to see if the silver nanoparticles anyway were toxic by themselves. So what we are checking in bacteria though is the one where we are combining silver nanoparticles with methylene blue, and it also has the irradiation with the red light. They're using that. So there is a three-way effect on the bacteria, which we are really not measuring with the HEC 293 T cells, because we were only looking for the toxicity. Now we did look at the methylene blue toxicity by itself, and they were not at the concentration that we are using. They are not toxic, by themselves. We did not really try the combinations, and we can, but we didn't really try the combinations because we were not sure if it'll, be of any significance. But that's something we can just to see if together they won't be. So I think the effect mainly is in the presence of the light here, the toxic effect.
Well, I mean,
Go ahead, please.
Oh, well, I mean, I just thought maybe you'd treat them the same. I mean, I assume you would, what's the hypothesized route of using these? You inject them into the joint and then everything would be exposed to the same.
Yeah. Basically, inject into, let's say, the knee in the synovial fluid and then irradiate it with the light. And then, of course, eventually that's one of the major problem of the PDT. I mean, this, the chemical is going to kill healthy cells and also bacteria as well.
Oh,
but
okay.
in the literature, we know that so they do preferably kill the tumor cells and the bacteria because they do have like different oxidation states. So I mean, yes, some of the healthy cells will be damaged as well, but that's in the PDT, that's not really the concern. Yeah, of course, I mean we need to minimize it, but when we do this cytotoxicity measurement, I mean we have not irradiated with the methylene blue. Otherwise,
Okay.
it'll also be active. Yeah.
Okay, I get it. I thought maybe there was something very different about the bacteria, how they took it up. Any other questions before we move on? Okay, let's see. Sorry, I lost my, darn it. I lost whoever's next. Whoever's next, please jump
It's
on.
me, it's Daniel. I'm trying to share my screen.
I had it up and then
there
I was like, yeah.
Or are you guys looking at the shared screen?
It says your speaker view, so yeah.
It always pulls up the wrong one for me.
Yeah, I know. Okay, thanks.
All right, thank you. So yes, I'm Daniel Scott, and I'm at Center College in Danville, Kentucky. I'm in the Department of Chemistry, and so I'm excited to share some of the work we've been doing to try to make new cancer therapies with some of the undergrads here at Center. So. Change the slides now. There we go. So again, ultimately our goal, we have two sides of the project that are going to come back together at the end for the common purpose of trying to alleviate a lot of those non-specific side effects and a lot of the non-specific interactions you see. And a lot of times they're associated with cancer therapy. So you think about suppressed immune system and losing your hair and all the side effects that come along with chemotherapy. We're trying to negate some of that by two different avenues that we're really trying to approach this through. First avenue is trying to make better drugs, essentially make drugs that are going to interact specifically with the cancer cells and be a little bit more toxic to cancer cells, with the idea that we can give less drug total, so we have less side effects, but still going to kill the cancer cells. And then the other type of avenue or approach that we're going to take is try to better deliver those drugs to just the tumor tissue, right? So we're not going to have a random distribution of the drugs throughout the entire body. We're trying to focus in where we're delivering those drugs so that they only end up into the tumor tissue. And so that way we're not going to have all the side effects we see with the normal healthy tissue. So the first part, again, we'll talk about is with a natural product drug called Mithramycin, and then we'll talk a little bit about using gold nanoparticles to try to deliver the Mithramycin and other anti-cancer drugs after that. So the anti-cancer drug Mithramycin is an older drug. It is originally used in the 60s for anti-cancer capabilities, but was abandoned due to a lot of the side effects that it had. But it was pretty toxic and it has a lot of unique functionality. And so what we're trying to do is to try to modify this drug and make it better moving forward. So there's about 25 different enzymes that put this drug together in the bacteria. It's originally found in the soil bacteria. And then one of the modifications we've made is to inactivate the last enzyme in that biosynthesis. And when that happens, it actually causes the accumulation of these three different Mithramycin analogs. And so everything is exactly the same on these structures other than the side chain region of the molecule here. So we have Mithramycin SK, SDK, and then Mithramycin SA. And so the reason we're interested in these three derivatives is because the Mithramycin SDK and the Mithramycin SK analogs showed increased anti-cancer activity. So they were quite a bit better than regular Mithramycin in terms of cytotoxicity towards the cancer cells. And the only place that they're different is on this side chain in the third position up here. So we know through some other work that this side chain is associated with interacting with the sugar phosphate backbone. Mithramycin binds DNA. It binds GC-rich DNA and cross-links it together. And so again, the way it's working is shutting down transcription of several of these proto-oncogenes that are upregulated in different cancers. And so it's not specific for cancer, but these genes are upregulated in cancers. And so we're trying to turn off these processes selectively in the cancer cells. So this part of the molecule, again, is shown to be very important for the activity. And there's only so many natural derivatives that you can get through the biosynthetic pathway. So I mentioned SDK and SK are both better at killing the cancer cells. The other analog that is produced, and also all three of these accumulate together, you can't get it to selectively produce one over the other. But the Mithramycin SA showed a much decrease in anti-cancer activity. And so part of the speculation in terms of why that is, if you look at this functionality on the side chain, the carboxylic acid, at our physiological pH, that carboxylic acid is going to be deprotonated and have a negative charge on there. And so speculation is that when it's trying to interact with the sugar phosphate backbone, the negative charge on that sugar phosphate backbone and the negative charge of our carboxylic acid are not really going to like each other. And so trying to get rid of that negative charge from the carboxylic acid, but also add in some new functionality. This is the only carboxylic acid in that molecule. We've decided to try to target that location to add on some new functionality and new abilities to interact with the DNA. And so what we do is we grow the cells, we isolate the cells and lyse them open, and then we collect our three different drugs, and then through some different chromatography methods, we will isolate our Mithramycin SA as our starting material. And then through just a one-step reaction, you can think similar to a peptide coupling reaction. What we do is we take another compound with a primary amine and we couple it to that carboxylic acid. So in that process, we are not only getting rid of the carboxylic acid, which again, speculating is what's causing some of that decrease in anti-cancer activity. We are also adding on some new functionality to that side chain and looking to see how that's interacting with the different cells and the DNA in there. So we've been able to create a little library of different compounds. And what we do after we get the compound is we use the A549, it's a non-small cell lung cancer cell line, just grow those in a dish. And then similar to the MTT assay that our first speaker talked about, we actually use a residuant assay, which is similar just to measure cell viability. And so we add different concentrations of our drug to our cancer cells, and we're essentially measuring how many of the cells are decreasing that metabolic activity. So what was exciting for us is through this, a lot of the first analogs we tried had some smaller side chains and just, again, trying to get rid of that carboxylic acid. But through the process, we tried some aromatic amino acids, the tryptophan and the phenylalanine derivatives actually showed really, really good activity. And so again, measuring our IC50 values, lower IC50 value means it's more toxic to the cell. Comparing our Mithramycin SA, again, took a lot of drug to get to the point where we're actually killing some cells. Then we're measuring against our SK, which is our previous gold standard in terms of the different derivatives of Mithramycin we've seen. We're actually able to increase it quite a bit, increase the activity compared to the SK with both the tryptophan and the phenylalanine derivatives. And then that was our first set of aromatic compounds we tried. And so then from that, we actually tried in some new derivatives. These are labeled compound 1 and compound 2 right now, because we're still trying to completely characterize and make sure we have the actual structure we think we have. But both of these compounds have a tetrazole ring in those, which is a 5-membered ring with a couple nitrogens in there. And so we were pretty excited to see some even more increased anti-cancer activity with these different derivatives. So again, building off of this, our next step is to, again, think about these molecules that have worked in terms of the modifications, and can we further specialize that for our cancer cells and increase that activity towards the cancer cells with that modification on that Mithramycin side chain. So again, that's the first part, is trying to increase our anti-cancer activity through just making better drugs. The second part is trying to better deliver those drugs. And so what we're using to deliver our drugs, you think about a lot of times when you're injecting these cancer drugs, it may be through an IV infusion. And so we're going to try to use a nanoparticle delivery system to selectively deliver our drugs to the tumor tissue. And so that selective aspect comes from what we're going to use is called passive targeting. So as tumors are growing and as they're rapidly mutating and rapidly expanding, they create somewhat of a leaky vasculature and has some irregular shape to it. And so this leads to what's called the enhanced permeation and retention effect. And so your normal cells, your normal tissue, the cell lines are packed pretty tightly together, where nanoparticles are too big to get in to our normal tissues. But the cancers, where they're growing again very rapidly and have that irregular vasculature, they actually have some pores and some gaps in there that the nanoparticles can actually selectively accumulate in just through a passive targeting again. So they'll circulating through your system, and when they get to the tumor tissue, they can just fall through the grates you can think about and end up in the tumor tissue. And then once they get in there, they don't get removed very well. And so our goal is that if we can package our drugs in the nanoparticle delivery system, that they will selectively accumulate in the tumor just based on the size. And then ultimately what we're going to try to do is to remotely activate our nanoparticles to release our drug once we've seen that they've accumulated inside the tumor and given them some time to accumulate in the tumor. So building up our nanoparticle that we're going to use to deliver this. So again, I mentioned Mithramycin is a DNA binding drug. And so we're going to try to take advantage of that DNA binding to load and deliver our Mithramycin drug and again, other DNA binding drugs in the tumor tissue. So the first part of our nanoparticle is going to be a gold nanoparticle core. And so we chose gold for a couple of reasons. One, just by itself, gold is relatively inert, and so it's not going to interact and have a lot of toxicity by itself in the physiological systems. And they're easy to functionalize in the nanoparticle or nanometer size range. And so we can get a range of sizes of our nanoparticles from 1 up to 100 nanometers pretty easily just by modulating how we are synthesizing those. And then they're also relatively easy to functionalize on the surface. And so if you have free thiols in solution, you can form a gold thiol bond on the surface of that gold nanoparticle. And so we're going to use that chemistry to try to decorate the surface of our nanoparticle to load our drugs, but then also try to make them relatively evasive to the other physiological components in your system. So they're not going to get cleared really quick and have a lot of non-specific interactions in the body. And then lastly, gold nanoparticles will actually absorb near infrared light. And when they absorb that light, so those wavelengths, you're thinking 800 to about 1,000 nanometers in wavelength. And those wavelengths tend to be relatively low absorption from other biological components. And so you can get a little bit of tissue penetration with those wavelengths of light. But when the gold nanoparticles absorb that light, it actually causes them to heat up. And so what we're trying to do is use that generation of heat when we shine that near infrared light on the nanoparticles to cause our nanoparticle to heat up, and then use that heat to trigger the release of our drug in solution. So other components on our nanoparticle surface, we haven't got to the point of actually putting this on yet, but we're going to eventually use some polyethylene glycol. And so that will be to try to make our particles stealth once it's injected into the system, into the biological systems, where it's going to avoid some non-specific protein interactions and allow it to circulate a little bit longer through your system. And then we're going to, again, through that gold thiol bond, we're going to label the surface with a single-stranded DNA sequence. And then we are going to add in the complementary sequence to that DNA sequence and just have it base pair and create that double-stranded DNA sequence that our drugs are then going to recognize. And so once we have our double-stranded DNA sequence on the nanoparticle surface, we'll load in our drugs, and those will interact with that DNA, double-stranded DNA, and basically attach to the surface of our nanoparticle with the idea that then we'll be able to inject those into the system. And so ideally, we're going to be able to deliver 2 different drug payloads. And so the idea here is that you can have some combinational therapy. I mentioned Mithramycin was used a long time ago and was since abandoned, but there's been some more recent work showing that in use in combinational therapies, it's actually shown to be pretty promising. So the idea is that you can deliver one drug early on, and then after that drug's had a chance to take effect, you can then deliver the second drug after that. So again, the idea being with our near IR, near infrared irradiation, that we will have both drugs loaded onto the surface of the nanoparticle. And then once it's accumulated in the tumor, we will irradiate with a certain amount of near infrared light and cause our first drug to release. And then after that drug is released and had a chance to work into the cells, we will irradiate with more near IR light and have our second drug be released and sequentially from that first one. So as a model for this, we're using, this is just regular Mithramycin, again, not one of our new derivatives, but thinking, again, in the end, once we get our new Mithramycin analogs, this will couple pretty well with our delivery system if we're already able to deliver regular Mithramycin. And so that's one of our drugs. And our other drug that we're going to use as our model system is called doxorubicin. And so this is a previously used anti-cancer drug. This was one of the actually first anti-cancer drugs to be incorporated into nanoparticles that had some heart toxicities associated with it. And so there's a liposomal formulation of doxorubicin that gets used clinically. And so these were our 2 model drugs. And again, is also a DNA binding drug. And so what we did is we engineered the DNA sequences on the surface of our nanoparticles to selectively bind to our anti-cancer drugs, but then we also modulated how strong that interaction was between the strand that's on the surface of the nanoparticle and that complementary strand, so that ideally we'll be able to irradiate with light and get these to release again sequentially in order as we shine more light on them. So first step was trying to choose what size of gold nanoparticle we wanted to use. And so we chose 20 nanometer particles because when we use that 20 nanometer size, when we irradiate it with our light, what we're looking at is just an increase in temperature. We got our highest increase in temperature quickest with our 20 nanometer particles. And then again, loading those onto the nanoparticle surface with our DNA and our drugs, what we're looking for then is, can we release these in different fashions based on how much light we're able to irradiate with? And so again, what we're looking at on the x-axis is how long we were irradiating with our near infrared laser. And then on the y-axis is the percent of drug that was actually released from that nanoparticle surface. And so again, as you can see from our plot, we got our Mithramycin to release pretty quickly. And so within about 5 minutes, we had the majority of our Mithramycin released from our nanoparticle surface. And then our other modifications with our doxorubicin bound. Ideally, what we were shooting for is 3 different release profiles. But ultimately, when we saw 2 different release profiles, I don't think these 2 with our different dox formulations are significantly different from each other, but both of those showed more of a delayed release from our nanoparticle surface after irradiation with light. So next steps with this are to try to actually put our new Mithramycin drugs that we're making in our nanoparticle and release that, but then also do some cytotoxicity assays with our cancer cells and see, again, what the toxicities look like with irradiation with our near infrared light. So with that, I want to thank the talented undergraduates I've had a chance to work with over the last couple of years. On the nanoparticle side, so Cole Hilt, shown here, and Makai Benford did a lot of that early work with Gracie picking up some of the work this last year, and then Daniel Lowe, who's, I don't have a picture with him in here, was able to pick up with that and is continuing on with that. And then Evan Miller and Kate Jenkins did some of the foundational work with the new drug, Mithramycin drugs, and then Maddie Jenkins and Mary Elena Blairanne have picked that up more recently as well. So with that, I'm happy to take a couple questions and answer anything you have. So thank you.
That's great. There's somebody's, well, just a comment in the chat. I think this is really cool, and it just made me think there's this guy, Marty O'Toole at University of Louisville.
Okay.
Have you ever met him?
I have not.
Well, the only reason I know about his research is because he was using the EM core to look at gold nanoparticles in glioblastoma cells. So they're doing, some things. I mean, I might introduce you to him, and
Yeah,
seems like
great.
doing kind of similar kinds of things. I mean, he's in engineering too, so it's not a.
Okay.
So anyway.
Yeah, that'd be cool.
Just a comment.
I have a question and comment type. So, I mean, I just want to make sure that, I understand this correctly. So normally I know that, the people, they use like, the different ligands so that they can selectively bind to the tumor, and then they can modulate the drug release. But you don't really functionalize the gold nanoparticles, right? So you basically count on the size of the nanoparticles, basically enters into cell through the leakage. Is that right?
Yes, that's correct. So you can add some active targeting. So again, that polyethylene glycol that was on the surface as well. Right now, we're trying to use that just to avoid recognition, but that's a common place if you want to do some active targeting to target tumor cells and look at overexpressed surface receptors and things like that. You can help get some increased specific targeting as opposed to just the passive targeting based on the size.
Okay. So that PEG, does it basically act like the stabilizer? Does it prevent the aggregation? Or does it just help, to diffuse through the cell?
So the once we actually add it into the physiological system, it'll actually help it'll help avoid non-specific protein interactions on the surface of that. So you're not going to get cleared. It's not going to get cleared quite as quickly. So the DNA, it can help prevent aggregation just between the nanoparticles. Once we get the DNA on the surface of the nanoparticles, those do a pretty good job also of keeping the nanoparticles from aggregating together. You just get some charge repulsion on that surface.
One more thing. So normally, I know that this, the photothermal therapy of this gold nanoparticles, they have size dependency. To my knowledge, though, the preferred size was around like 50 nanometer, but in yours, it was like 20 nanometer. I mean, is that kind of how to say? I mean, how is it different? Why is it different than the usual size? Is there any special reason? Or
some of it probably has to do with the wavelength of light, the actual specific wavelength of light you're using. So the absorption of that light will change with the nanoparticle size. And so our particular laser liked the 20 nanometer particles best, but again, choosing a different wavelength, you may get much better absorption to a different size.
I see. Thank you.
Okay, great. I think, sorry, Lindsay, we're a little behind, but don't worry, we'll just keep on going a little after.
That's okay. Can everyone see my slides?
Yeah, looks good.
Okay, thank you. First, I just wanted to thank Embray for kind of putting this platform together. It's very, very neat. All of us researchers across the state with very similar research interests getting to talk. I think it's a very, very nice thing that has been created. And secondly, I went ahead and I modified my presentation really quickly while we were sitting here listening to the other researchers because Dr. Williams did a great job in the beginning talking about platinum and the background. And so I quickly removed that, and that will help us with the time constraints. And just like Dr. Scott talked about the idea of designing drugs, mine's actually an active design. And so we're using hormone LHRH to target an LHRH receptor. And on this slide, this is Dr. Ningiri. She's my collaborator out of chemistry and myself. And so I will talk a little bit about how we kind of designed these compounds and what our main goals are for them. First off, my drugs are towards cancer. We are looking at reproductive cancers mainly because they overexpress the LHRH receptor. We're focusing a lot with women health issues. I have on here, women are 4 times more likely to die of a reproductive cancer than a car accident, right? And us women, we get kind of dinged for saying we're bad drivers. But in reality, we should be more concerned with our cancer rates. So we have about 500,000 people afflicted with reproductive cancers each year. That's ongoing, and 50,000 of them get breast cancer in the range of triple negative, which is one of the ones we're most concerned about because it's highly aggressive and invasive, and you have about 8 to 13 months of survival after you have been diagnosed with this cancer type. Now, there's one FDA drug that has been approved within, I think, the last 6 months, which is exciting, and it's a conjugated drug, which is very similar to what we are trying to propose here with ours. Now, just a little bit of background. I kept one slide in. This is showing you the cisplatin and carboplatin that was previously talked about in this platform. But I'm left it in because it talks a little bit about the side effects of the physiological systems. So the cisplatin, which is a very smaller type drug, is highly lethal. Its LD50 is extremely low. And so in some cancer types, it's used as an end stage drug. And it has high neuropathy, high nausea, it suppresses the immune system. So we get co-infections a lot of times in these patients, which is something that you never really want to get. And the other case of the drug that's commonly used is carboplatin. We talked about previously that it's a little bit bigger and bulkier, which is great. It reduces the side effects. However, it's not as efficacious. And so you need to use actually a larger amount of this compound to get very similar type suppression of the tumor, which you'll see with cisplatin. So there are benefits to carboplatin, but there's also some not so great things dealing with that drug as well. And so one of the very first things that we tried to do was not just go after suppressing the tumor, but actually diagnosing it. So we're coming up with a theradiagnostic approach, which is a diagnostic approach and then also a treatment approach, both of which are targeting an LHRH receptor. And so this is a radionucleotide that we're developing where you've got the radioactive metal and we designed the linker and then we added LHRH to it, which will drive it towards the cancer cells that are overexpressing that LHRH receptor. Now, the idea of this radionucleotide is we use an SPECT or PET scan images so that we can find smaller localized tumors earlier, things like that have metastasized, or just diagnosing the primary tumor itself, but we'll get a more crisp and precise image. So in the first panel here, this is just showing uptake of our compound. So this is rhenium alone, and this is rhenium LHRH tagged, which is ours. And so we get higher uptake into these triple negative breast cancer cells. And then B is an MTT assay that we've all, I think, have used before that some of our speakers have said, and which we're showing that there's no toxicity associated with our LHRH, which is wonderful, meaning it's able to be cleared and it's able to be metabolized within the patient. Now, once you diagnose a patient, it also will tell us that it does express the LHRH receptor, this cancer cells. We can quickly then use a targeting chemotherapeutic. And one of the things that I did want to kind of express is the earlier that you can diagnose a cancer, the survival rates of those patients are much higher. So if we can diagnose it earlier, that's really excellent. And then if we can give a targeting therapeutic, that enhances the survival rates of these patients. And so in this diagram here, instead of the radionucleotide, we have our platinum compound, and we added a linker and the LHRH molecule. So a very similar conjugation. And here what we're showing is we have our tumor growth model. It's an in vivo model where we have normal control, which is the diamond. And then we have this group of lines right here. This is all carboplatin, various doses from 2.5 up to 20. And then all the lines on the bottom are our drug. So 2.5 all the way on up to 20. And this is in 4T1 mammalian cell line that was implanted into the animals. And this is by an IV administration. So we see tumor suppression with our chemotherapeutic compared to carboplatin. Now, next we did this again in another model system. We did this in a human MDA cell line. We just didn't want to do one cell line. We wanted to do a second to show that it does work. And so in this one, we did a different dosing regime. We only did 10. And we did 10 because we wanted to bring in the drug cisplatin. If we used any higher doses of cisplatin, we're getting into that LDL 50. And so we didn't apply cisplatin in the previous model. And for this one, we don't see as big of a suppression as the previous one, but these cells, they don't proliferate as aggressively as our prior 4T1. So this is something that we thought and we knew we would expect. But one of the great things that we do see is it's working better than both carboplatin and cisplatin, which are shown right here. Now, in the lower panels, A, B, and C, what we've done is we've taken out tissues out of these animals. And in panel A, we see our platinum drug is getting more targeting and uptake in the tumor compared to the skeletal muscle. And if we compare this between tumors, we can see more of our platinum drug gets in compared to carboplatin within the tumor tissue. And then you can see here in panel C that there is a lack of targeting dealing with, with carboplatin because it's about similar in the amount that gets into skeletal muscle that does not overexpress the LHRH receptor. Now, this is showing tumor growth to 1500 volumes. So this was the end point that we said for these animals. The green line is showing you carboplatin. The red line is showing you cisplatin, and this is our drug, Platinum-LHRH. And so we were able to suppress the tumor to a greater degree than both of the other two compounds. And we were starting to see toxicity with the cisplatin group. So we're able to show a safety profile and also hopefully an efficacious profile dealing with our compound. Now, one of the things that we did want to point out was in designing our compound, the linker was actually a really wonderful design in which it can be cleaved by an esterase and it can revert back to actually cisplatin. So our overall goal is when we made our compound, we made it to look similar like carboplatin. So when you inject it by IV, you get less detrimental effects, right? Because we said carboplatin is the safer one. But however, when it gets inside the cell, the linker is able to be cleaved. And when it's cleaved, we're hoping it would revert back to cisplatin. And so this graph up top is showing you our compound with esterase, and it was able to be cleaved, and it shows you that it reverts back to cisplatin. And so we have made a prodrug, which hopefully is a lot quicker to pass through FDA approval with using our drug reverting to cisplatin. And down here in this panel is just showing you cisplatin as well. And so with that, I'll take any questions. I did shorten my presentation a little bit, and hopefully that helped. Thank you.
Thanks, Lindsey. That's really exciting. Anybody have any questions? So you're doing all this animal work at EKU. Do you have the animals with the tumors there and everything?
We did. We did this first modeling. This was at EKU. The second one, we were able to get a grant for a third-party validation, and that was what validated our first panel right here. But then the lower ones we've done with collaborations out of UK.
Nice, nice. It's very exciting. Well, I guess you kept us almost on track. So
I did good.
Yeah, thanks everybody, and hopefully you guys can all communicate more after this, and we'll have this, of course, the recordings on the website. So thanks everyone.
Thank you.
Thank you.